Welcome to the future, where you will have tailor-made babies. Whether it's their gender, the tendency towards a psychological or physical skill set, it's yours to decide. There will be humongous crops, as well as cures for diseases which are unable to cure right now. This and even more will be possible with the revolutionary gene editing tool, the CRISPR Cas system. CRISPR Cas is a system which is used to cut DNA with high precision at predetermined location. It was first found in bacteria where it's used as a kind of immune system against bacteriophages. Bacteriophages are viruses infecting bacteria cell and hijacking them for the production of more phage. So let's take a closer look. The phage does this by attaching to the bacterial membrane and injecting the DNA into the bacterial cell. Phage DNA is incorporated into the bacterial genome and the bacterial cell is now producing new phage. When the number of phage reaches a critical point, the bacteria gets destroyed and the phage are released into the environment. However, before this can happen, a Cas protein will take the DNA and cut it, so it's innocuous. The protein is able to do so by recognizing the phage DNA with the help of the cRNA, which fits perfectly to the sequence of the phage DNA. So as soon as the right DNA sequence is entering the cell, the CRISPR Cas system recognizes the sequence and binds to it. In the process, the DNA unwinds and is cut. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Short palindromic repeats are small sequences which, when read forwards and backwards, result in the same, like Mr. Owl ate my metalworm. The sequences interrupting the palindromic repeats are basically bacteriophage DNA from previous infections. These pieces are stored in the CRISPR sequence like a library. Is the phage now trying to infect the cell again, the Cas protein is able to identify the phage DNA with the help of the cRNA. Cas means CRISPR-associated genes. These genes contain the blueprint for the protein. The resulting Cas protein is an enzyme, a so-called endonuclease, a protein able to unwind and cut DNA. And this is the reason why this system became so famous. The combination of a protein which can unwind and cut DNA with high precision, predetermined in the lab, results in a sensitive and specific altering of DNA. Moreover, the costs of gene editing are reduced by the factor of 10. The possibilities to use the CRISPR Cas system are manifold, from switching off genes to put in tailor-made sequences. Moreover, it's possible to modify the Cas protein, for example with different fluorescence dye, to mark specific DNA sequences which can help to check a patient's DNA for sequences that could indicate a disease. However, will we be able to have our own designer babies, gigantic crops, or even a cure for a bunch of diseases? We don't know yet, but with time the potential applications of the CRISPR Cas system surely will rise.